So let's move to the next talk by Blanca González. Blanca is doing her PhD with us at the Center for Biomedical Technology of the Universidad Politécnica de Madrid. She studied materials engineering and industrial mathematics, and now she's working on cell mechanics and cell biophysics. So Blanca, if you are ready, you can start now.
Yes, thank you. Okay, so hi all, it's Blanca up here. I would like to say thank you for the opportunity to talk today, since it's my last year of PhD, and well, I think it's a good training to learn how to compress data within few minutes for the thesis defense. So the topic of today's presentation actually represents one of the major projects that we have developed through my PhD thesis, in which we try to evaluate the contributions of immune cells, biomechanical features in immune dysfunction and aging. Okay, so first of all, when we talk about age, for example, we typically use the concept of chronological age. So in my case, I would say, my chronological age is 28. And it is true that biological malfunction and increased risks of pathologies come with this chronological age. However, the term chronological age is also misleading because there is remarkable variation in aging rates and expected lifespans within individuals of the same chronological age. So instead of the concept of chronological age, there is this other concept, biological age, which is rather used as a better indicator of health condition, remaining healthy lifespan, and life expectancy. So biological aging is usually explained, probably you all know this diagram, as the byproduct of molecular and cellular changes that can be considered as hallmarks of biological aging. So rather than reading this whole list of hallmarks, because any one of these could cover several presentations, I just introduced the one related to this work, which is immunosenescence. So immunosenescence refers to the gradual deterioration of the immune system brought on by the natural aging process. Immunosenescence involves both the host capacity to respond to infection in a worse way, and also the development of long-term immunity, immune memory, especially by vaccination, which also deteriorates as we age. We can see in the diagram, for example, how when we become elderly, we have lower levels of naive T cells. We have a larger number of senescent T cells, and we also have larger numbers of inflationary T cells. Okay, so this age-associated immune deficiency or immunosenescence happens to everybody, both in long and short-lived species, such as us, Homo sapiens. And even in species that only live 2 years, we can also see this process as they age. One of the interesting features about immunosenescence is that besides the gradual decline in both the adaptive and the built-in immune function, it appears the process called inflammaging. Inflammaging is the chronic low-level inflammation. It is the underpinning, inflammation is the underpinning cause of most of chronic diseases of aging. So if we want to treat our bodies in a way that we won't have any of the diseases associated with aging, such as diabetes, cholesterol, heart diseases, or arthritis, the common thread of all of them is inflammation. And somehow also it is interesting to research immunosenescence as a direct cause associated to this inflammaging process. So the motivation of our work enters in this part of the presentation because as biophysicists, we also wanted to include in the frame of immunosenescence the biophysical features of cells, in the sense that there are several relations between cell states, right, and biophysics of the cells. For example, very clearly in the case of immune T cells, it's the function of T cell migration. Here in the diagram, we have a leukocyte extravasating blood vessel. And for this particular function, the cell has to deform through the interstices. And for this, a proper set of mechanical features is required. Now in the vivo video that you can all see in the presentation, well, we have this permanent migration of T cells in red moving through endothelial tissues. So the aim of our project was to evaluate the biophysics scenario in the age-associated changes of immune T cells. How did we do this? Well, we designed a longitudinal experiment study using as animal model mice, and we performed several characterization. Excuse me. We performed several characterization techniques at 4 age groups. So first of all was the young adult groups at age 40, around 40 weeks. Then at the mature age, when the individuals were 56. Then at the old age, at 72 weeks. And then when they were very old, around 96 weeks. This was in collaboration with the group of De La Fuente at Universidad Complutense de Madrid. So then for each time point that I just showed, we performed the following experiments. So the cells were extracted from the peritoneum of mice, and then the part of the global populations of these cells were placed in Boyden chambers, because we wanted to see how functional, in the sense of how these cells were able to spontaneously migrate through pores. Then with the remaining global populations of these cells, we sorted our cells in four cell populations. We investigated CD4, CD8, both of them in naive and memory states. Once the cells were sorted, we performed aspiration tests using micropipette aspiration. Then with part of the sorted cells, we performed fluorescent immunostaining, and half of the cells were set in glass slides so that we could see the morphology of them, and half of them were put in cytometry tubes so that we can analyze some proteins of interest traditionally related to the migration and mechanical properties of T cells. So we had, like, in the end of each time point, these four aspects of the biophysical landscape of T cells. Then, yes, for you to have an overview of the type of experiments we perform and the data we obtain regarding the functional assay, you can see in the images how the Boyden chamber is covered of micropores of approx the same size of the cells. And when we move up with the microscope, we can see in the lower part of the chamber those cells that have been able to migrate through the pores. So in this way, we can, for example, assess the density of cells per experiment that have been able to migrate through the pores. Regarding the deformability, this is a typical example of a micropipette aspiration experiment. We see the whole cell being aspirated as we decrease the pressure, the differential pressure inside the pipette, and the type of graphs that we obtain are automatically processed, and we can obtain parameters such as the elastic modulus, the apparent elastic modulus, the apparent viscosity, and the variation, the coefficient of variation of these parameters per experiment. Regarding the internal composition experiments, we are provided with the information of the flow cytometry in a way that you can see here. We can represent, for example, protein of interest, expression of protein of interest versus forward scatter light or whatever other parameter, and different parameters or features of the proteins of interest that we were investigating can be in this way quantified. For instance, we looked at actin, myosin, bimentin, 5-methylcytosine, which is associated with the condensation of the DNA itself, cell fluorescence by lipofuscin molecule and the relative frequency of sorted cells, so that we can check whether there are more CD4, CD8, or naive or memory state. And finally, regarding the morphology experiments, well, in the confocal, we get images such as the one that you can see by performing thresholding, segmentation, and if we want to measure, for example, relative size of the nucleus, we perform an ellipsoid fitting, or if we want to see the actin cortex, we perform other type of Gaussian fit through the cortex. And overall, we have a set of morphological indicators of actin, DNA, and lamin B1. So now, once we have all the experiments performed, after the longitudinal study finished, we move to the analysis of the data, first by the basic quantification of the biophysical parameters that I just explained in the first type of characterization experiments. Then we wanted to see how these parameters were correlated with age. So we performed a correlation analysis. Then we also wanted to investigate whether there were more or less similarity between, for example, T cell types, CD4, CD8, memory naive, or parameter class. We are talking about mechanical parameters, morphological parameters, etc. Finally, we also wanted to investigate whether there were more or less similarity between individuals, or if this difference was not existent. So regarding the statistical tools that we use to investigate the correlation with age, well, we had the parameter listed according to their class, and we performed this kind of Spearman correlation test through which, well, using a regression model, we can estimate the extent to which the correlation, the linear correlation between the parameters and age is strong, moderate, inexistent. And the typical way, as you know, for doing this, for example, is to set a threshold value of P. And in this way, we can see whether there are some parameters that decrease with age, or some others that increase with age, for example. And then for the similarity analysis, the statistical tool that we have used for this purpose was the Jacquard index. So what we did was to represent the set of parameters, as you can see in the radar plot. So we have in each color age group, so young, mature, old, or very old. And then in each vertices of the polygon, what we have is one parameter normalized from 0 to 1. So the Jacquard index, what allows us is to quantify the intersection between the different polygons. So the ratio of the Jacquard index is given by the intersection of two sets divided by the union of the two sets. So 0 Jacquard index means that there is no intersection. 1 Jacquard index means that there is a complete overlap of the two sets. So in this way, basically, we can also create a heat map by investigating the Jacquard indexes of the different polygons that we are able to draw. So the first kind of, well, I'm going to show the raw data or the raw results obtained for the first step, which was to investigate how the parameters correlate with age. And well, we can see, let's say qualitatively first, some kind of conclusions such as that there is a significant age-related increase in mechanical parameters. For example, we observe in the 4 cell types that we investigated as cell stiffening, as cells age. We also found out that there are also changes in the sorting relative frequencies. For example, the ratio CD4 to CD8 decreases with age, and the ratio of memory to naive cells increase with age, as expected in this way. Also, now regarding the internal ordering given by the protein levels, we, for example, found that lamin B1 decreased with age, that also 5-methylcytosine decreases with age, and that self-fluorescence of cells increase with age. And finally, regarding the functionality, we also appreciated a loss of spontaneous migration in our experiments. Regarding the similarity analysis, well, we managed to display all the data, as I just commented, to evaluate the Jacquard indices of similarities for each T cell group. No? So we draw this kind of plots for CD4, CD8, memory and naive state, and we compare polygon versus polygon in order to have a Jacquard index. So the results are here and need to be further processed because we now want to move to the questions of, are there similarities, for example, between age groups? Are there similarities or difference between individuals? And this is an ongoing work, so that's why I prefer to simply give this general overview of our results, and hopefully you can all come to my thesis to see the final results. And well, so the conclusion of my presentation, now, as I explained in the introduction, we wanted to include the biophysical features in the landscape of the immunosenescence biological process. So far, no, regarding, for example, mechanical features, we see differences significantly correlated, for example, of stiffening of cells with age. Regarding functionality, we also have found decrease of spontaneous migration. Regarding internal ordering, well, two examples are lamin B1 and 5-methylcytosine, and we want to correlate this protein and molecules with, for example, the loss of nuclear compactness that we also found in our morphological studies. And where regarding the similarity analysis, we for the moment found that there is less similarity among aging groups and cell types. And as I said, this is ongoing work, so in the future we want to further analyze our results to investigate in individual variation and to combine all the parameters in order to perform cluster analysis as well. And this is all. Thank you to our collaborators for sure and the funding opportunities, and to all of you for your attention. And if you have questions, I guess in 20 minutes we'll answer them.
Thank you, Blanca, for the nice talk. So again, we have some time for a quick question or a couple of questions now. Or otherwise, we can just move to the last talk, and then at 1:20 we'll have time for the questions. So yes, thank you again, Blanca. And the next speaker